In the fall of 2018, a single cell lining the human intestine was found holding a direct, synaptic wire straight into the base of the brain, and neuroscientists still cannot explain how that one connection hijacks decisions made nowhere near the stomach. The discovery came out of Duke University, in the lab of Dr. Diego Bohórquez. He'd spent over a decade convinced the gut wasn't just plumbing. Most researchers treated it as a bag that digested food and sent slow chemical signals upward over minutes, sometimes hours. Bohórquez didn't believe the timing worked. Depression, sudden cravings, gut-level dread, these hit too fast for a hormone drifting through the bloodstream. He wanted to find a faster wire. In 2018, using a rabies virus as a tracer, his team found one. A specialized gut cell, called a neuropod, physically synapsing onto a vagus nerve fiber. Not signaling nearby, touching, wired in like a nerve cell talking to a nerve cell. Here's the first anomaly. The team clocked the speed of that signal, gut to brainstem in under 100 milliseconds. That's faster than most reflexes, faster than the time it takes you to blink. For a century, the assumption was that gut-to-brain communication moved on a hormonal timescale, slow, diffuse, forgettable. This measurement broke that model instantly. A sugar solution touching the lining of a mouse's intestine reached the brainstem before the mouse had finished swallowing. Something in your gut was talking to your brain on a timescale usually reserved for pain or fear, or the yank of a hand off a hot stove. And nobody had mapped a reason for a digestive cell to need reflex speed. The obvious theory: this is just about hunger. A fast circuit for telling the brain, stop eating, you're full, makes evolutionary sense. Starvation kills quickly. A slow, sluggish appetite signal would be a liability. Researchers folded the neuropod discovery into existing satiety science and moved on. Case closable. A fast nerve for a fast survival need, food regulation, nothing stranger than that. Except that explanation collapses under its own data. Neuropod cells don't only fire in response to nutrients. They respond to gut bacteria. They respond to inflammation. They respond to compounds your microbiome produces that have nothing to do with hunger signaling at all, including serotonin precursors. And that's where it gets harder to explain away. Roughly 90% of the body's serotonin, the chemical most linked to mood, is manufactured in the gut, not the brain. If neuropod cells are wired directly into the vagus nerve, and the vagus nerve terminates in brain regions governing emotion, then a bacterium in your intestine may be pulling a lever connected to how you feel in under 1/10 of a second, with no hunger involved at all. Now the deepest part of the case. In follow-up studies, researchers altered gut bacteria in mice and watched behavior change within days, anxiety-like behavior increasing or decreasing based on which microbes were present. Severing the vagus nerve reversed those behavioral shifts, even when the same bacteria remained in the gut. The behavior wasn't coming from the bacteria directly. It was coming from the wire. Something living inside your intestine was producing a molecule. That molecule was touching a neuropod cell, and within 100 milliseconds a signal was arriving at the exact brain structures responsible for mood and threat response. Structures with no idea the message originated from a microbe, not a memory, not a real threat. Your brain was reacting to your gut as though it were reacting to the world, and it had no way to tell the difference. Scientists tried to contain this. Some argued the effect was minor, a background hum beneath the real, psychological signals from trauma, environment, thought. Others proposed it was one-directional top-down stress from the brain altering the gut, not the reverse. But germ-free mice, raised with zero gut bacteria, dismantled that argument. Colonize a germ-free mouse with microbes from an anxious donor mouse, and the previously calm mouse begins behaving anxiously, despite an identical brain, an identical environment, an identical life. The only variable was bacterial. The signal still traveled the same 100-millisecond wire. To this day, nobody has produced a full account of how much of your daily mood, your sudden irritability, your inexplicable dread, actually originates below your ribs instead of behind your eyes. The wire is confirmed. The speed is confirmed. What's moving across it and how much of what you call yourself is downstream of it is still an open file. So the next time a feeling arrives with no clear cause, no trigger, no memory, no reason you can name, what if the decision wasn't made in your head at all?